What's up, YouTube? This is Sam from Tyke Schooling, and today we're talking about the cell membrane through which the transportation occurs, which means we'll talk about these points. We'll talk about transport through cell membrane, diffusion through lipid bilayer, and in transport through cell membrane, we'll talk about the diffusion, which is simple and facilitated. And we'll talk about the failures of ions to pass through the lipid bilayer and properties of protein channel, factors increasing diffusion through protein channels, active transport, and secondary active transport. First, we'll talk about the transport through cell membrane. While talking about the transport through cell membrane, we'll use this diagram as a way to guide us in understanding the topic. So remember, transport through cell membrane occurs in two types. Like there are two methods by which transport through the cell membrane as a whole occurs. So how it occurs, what happens when, for example, let me explain it this way to you. All right, let me use a yellow color. Consider this thing, for example, let me use a marker. For example, this is your artery through which the red blood is traveling, right? This is your artery and red blood is traveling. And around your arteries, for example, these are small cells. For example, this is a cell in your muscle, for example, highly attached to each other. So what happens, and the space between the arteries and your cell is called the interstitial space. This is the interstitial space. All right, so this is, we'll mark it with the yellow color. This space is called interstitial space. So what happens, first the molecule or any molecule travels to the interstitial space. Let me use a different color for this one. The first molecule travels through the interstitial space, and from here that molecule enters the cell through the cell membrane, and it enters the through cell membrane. And there are three ways. There are two main methods through which it enters through the cell membrane. The basic one is your diffusion or passive transport, which means it doesn't take any energy, that's why it's called passive transport. No ATP is utilized, so that's why passive transport. So number one is your passive transport. Number two is your active transport. So both are used, active and passive transport. And again, as we move further, the passive transport, which is, I'll write down with a P. The passive transport is further divided into two types. That is your simple diffusion. I'll put it this way: S for SD for simple diffusion, and for the facilitated diffusion, we'll put FD. All right. So FD, passive transport, divided into simple diffusion and what we call the facilitated diffusion. So there are two main methods through which the transport occurs. There's another method through which we can call that a third way of transportation. That is called the what is called the diffusion through the lipid bilayer, and this we'll talk about this through diffusion through lipid bilayer in the end. So these two types are these selective diffusion and facilitated diffusion. This is a type of passive transport, which is this one. All right, the active transport is not shown in this picture. We'll talk about that that active transport later on. So first we'll cover up this thing. So what is diffusion diffusion or passive transport? It's actually a random molecular movement of substance down their gradient concentration, either through opening in the cell membrane or in combination with a carrier protein caused by simple kinetic motion of molecule, which is called the simple diffusion or the passive transport. Here what happens, for example, the concentration of yellow molecules, consider this side, on this side, this is the outer side, this is the outer side, O for outer and I for inner. So this is inside the cell, for example, and this is outside the cell. So outside the cell, you can see that on this side, yellow dots are higher, which means there is a higher concentration or higher number of yellow molecules. So they will move against the gradient. That they move down because they don't want to stay up there because they are large in numbers. So they want to move to a place where there are little numbers, so they move against the gradient to this side, which is inner side. So that on the other hand, we have got yellow dots, a lot of here yellow dots. So again, they move from higher concentration gradient to lower concentration. So this is called, they move through the kinetic, they have got the kinetic energy through which they move, and they move against the gradient. So gradient work as the energy provider. You can call that energy provider. Gradient difference is causing them to move. So in this way, we have, through the passive transport, we have two types, which we have the simple diffusion, and another type is the facilitated diffusion. Through the simple diffusion, what happens? The movement of substance through opening in a membrane without binding to any protein. It doesn't bind to any protein. There's an opening. From that opening, it straightly moves inside the, what we call, inside the cell. So there is nobody binding them. They are not, the molecules are not binding to anything. They are moving straight opposite and inside, opposite to the gradient, from higher gradient to lower gradient. On the other hand, the facilitated diffusion occurs through carrier protein. This, for example, this is a whole protein molecule which is kind of getting this yellow dots. For example, there should be a box, small box, and the yellow molecules are going to attach to this box, and these yellow molecules, when they are attached, there is a transformational change occur, and they move on the opposite side, and they open on the inside, and the molecules are released. So this is why this is how the protein helps moving the molecules from the higher gradient to lower gradient. So that's why we call it the facilitated diffusion. The word facilitated means it provides a facilitation, or provide a means somebody is helping them, the protein helping them, they're facilitating them molecules to move inside against the gradient. So there is no energy there's no loss of energy again here. We'll talk about detail how this works in another video, but for now you have to just know that. So this is the type of your diffusion, or you can say the passive transport. Now we'll move towards the diffusion through the lipid bilayer. Consider this thing. Now we're done with this picture. Now we'll move to this thing. This is called, you can call this and again, one thing I have just missed, you have to know some examples of a facilitated diffusion and simple diffusion. In simple diffusion, we have got the oxygen molecule and a carbon dioxide molecule. They does not need anything, and they move easily. And in the facilitated diffusion, there are molecules called glucose and amino acids. You have to remember them. Glucose and amino acids are example of facilitated diffusion, and for the simple diffusion, they are example of what we call oxygen and carbon dioxide. Now moving to diffusion through lipid bilayer. So what is lipid bilayer? So this is a two-layer molecule. So we'll take one cell. For example, we're taking this layer out, the single layer. So this is made of two things: a head, which is hydrophobic, and tail, which is the head is hydrophilic and the tail hydrophobic. So they do not love water. They love water, something like that. So what happens in lipid-soluble substance can easily diffuse through lipid bilayer. They can easily pass because they are diffusible. They can easily pass through this. And for example, we have the oxygen, which easily passes. We have got nitrogen. We have got alcohol molecule, which easily pass through the lipid bilayer, and we have got the carbon dioxide. Water, which is although is not soluble, which is insoluble, can diffuse through lipid bilayer by penetrating it like a bullet due to its small size and high kinetic energy. You have to remember, sometimes they ask like why water passes through the lipid bilayer. All you have to say, because of its small size and high kinetic energy. Because of its small size, it can easily go through this. For example, these oxygen molecule, they are also small. Carbon dioxide, they are also small. So for them, it's easy to cross the cell membrane. Now we have to talk about why there's a problem, why there's a failure of ions to diffuse through lipid bilayer. What happens sometimes, you have to remember that these molecules always carry charges. Like some molecules have positive charge, some molecules have negative charge, some molecules are neutral. So what happens when again, and some molecular sizes also differ. Some molecules are small, some are large. So we are going to talk about two things why some molecules do not cross the lipid bilayer. We know that water molecules are attached to ions, and they are hydrated ions, and when they are hydrated ions, they become big in size. When they have a bigger size, for example, this is a small comparatively to this molecule I have just made. So this is larger, this is smaller. So for this molecule to cross, it would be very easy. But for this to cross, it would be very hard because of its larger molecule, larger size. But if I add some ions to it, again making it much larger, and it will be hydrated, it will be much harder for this molecule to cross. The second reason why molecules cannot cross the lipid bilayer because of the charges on it. Because there's a negative charge on the surface and positive on the inside. For example, there's a positive charge on the inside and there's a negative charge. Let me change the color for you. And there's a positive charge on the outside, and there's negative charge on the surface. Sorry for this one. Negative charge on the surface, and there's a positive charge inside. So what happens, positive and negative ions are repelled. For example, there is a negative charge ion try to come in, this negative molecule tries to come in, and here again we have the negative negative charge on the surface, so they both will repel each other and the molecules will get away. So that's why it's very hard to go inside. Again, when a molecule tries to get inside, even it's positive, for example, it's positive, it's attracted, and the positive inside won't let it to move over. For example, it can cross, if it's positive, it can cross this upper side, but cannot cross the lipid part of the lipid bilayer, and it have to go back, repelled back to the outer side. So these are the two reasons why some ions cannot cross the lipid bilayer, because of bigger size, and they are sometimes hydrated, and because of their charges. So if they ask an exam, you got to be clear about this. So in this part, we'll just talk about these three topics, and for other three topics, we'll move to the next part, and we'll catch you there.